Hi, Bianca. It's great to see you again. I was hoping you could introduce yourself and present your research in general terms.
Yeah, hi David. Nice to see you again. It's been a while since we met at USC. So my name is Bianca Costa. I'm a PhD researcher at the University of Southern California, and an environmental engineer by PhD and training. However, most of my research, or I've had a lot of intersections with environmental chemistry, given the nature of my research. It's funny, just offended, so a lot of ideas flowing into how to define my research topic, but I title it Addressing Contaminants of Emerging Concern in the engineering water cycle. And in particular, I focused on PFAS, which also stands for per and polyfluoroalkyl substances, which is a mouthful, and most people just call it forever chemicals because they're supposed to last forever, or that's the assumption we're trying to debunk. And yeah, so whenever you think about PFAS and how do they relate to the engineering water cycle, my research focused on understanding where are we right now in the engineered water cycle in terms of like where is PFAS degree of concentrations and ubiquity and variety of PFAS at the drinking water, and particularly we did a community engagement study that evaluated drinking water already at the point of distribution to assess what's the degree of PFAS distribution and levels in certain communities that might be highly or higher have a higher exposure level given that they rely on alternative in groundwater sources in Southern California, which is already an evolving project that I would love to talk more about as well. And then also the part of okay, what can we do to remediate this PFAS contamination? And I love biology, and most of wastewater treatment or water reclamation facilities relies on biology. So a lot of my research also looked into how can we develop biotechnologies or use emerging biotechnologies to explore whether biotransformation of PFAS is possible, though if you talk to people that sounds like crazy to think about, and also understand what is happening also at the water reclamation facility. How are PFAS distributing there, and how can that be mitigated if we have a better understanding of what is happening to them, especially because they're so tricky to measure. As if you talk to any PFAS researcher, there's more than, I believe, there's thousands of PFAS compounds. So it's not only the complexity of measuring them, but also of categorizing them and also of extracting them from different environmental matrices. And that's where chromatography, sample preparation, and all of the chromatography techniques come into play. And it helps us to expand what we know in terms of distribution and effectiveness of our novel or emerging treatment technologies.
And speaking of several matrices, I believe that some of your research is derived from EPA method 1633. Is that correct?
Yes, I talked about biology and how can we use biology to address PFAS. Most of this biology, biotransformation, or so there's many aspects of measuring PFAS, right? As I mentioned, it's very tricky to measure them. Getting them out of a matrix, it's hard. The EPA came out with this method 1633. I think it was in draft mode for a couple of years and people were still developing, and it was finally published, I believe, at the end of last year. And this method, it's meant to provide like initial or a starting point for alternative matrices to drinking water, which is the EPA 533 and 537. And I know them by heart because I've analyzed so many data with these other methods. So 1633 was developed to really give the research community and all other people that might be interfacing a possible incoming regulation to give them tools to do sample preparation, extraction, and detection using liquid chromatography, mass spectrometry for the most part, in soils, tissue, and I believe, and in wastewater for sure, which is what I'm most related with. So 1633, it's very interesting because like for as much as you can standardize a method, every matrix it's gonna be different, right? You're gonna have different matrix effects. You're gonna have different suspended solids quantities. If you're talking about wastewater, you're gonna have inhibitors from the process, right? So it's very, it's really, this is like the idea of how you can get started But at the end of the day, if you want to have accurate and good quality measurements for PFAS in particular, you're going to have to do some very intensive method development. And a lot of it requires extensive sample preparation. For example, whenever we do wastewater characterization of PFAS, yes, it's based on 1633, and it follows this standard procedure of solid phase extraction and then measured in liquid chromatography mass spectrometry, or LCMS for short. So a lot of that extraction procedure, that's what needs more iteration and more customization to your particular needs. It's typically, and this is outlined in 1633, you start with methanolic extractions. So you're trying to basically desorb this very highly organic or very nonpolar structures, which are the PFAS compounds, with that perfluorinated or polyfluorinated tail with methanol. And then you're trying to extract them from whatever matrix that you have, typically wastewater. It's very has a lot of suspended solids, so you need to really try to extract it from there. And then you do, after multiple sonication and centrifugation steps, then you use that methanol supernatant and then you blow it down to dryness. But it's not exactly that easy. I think you still have to add more aqueous phase because you want to keep most of your samples still in aqueous prior to SP. And then you take your, I don't know, I think it's up to 50 milliliters of your resuspended methanol extract and put it into SP. And then you follow like a standard for PFAS wax or weak anion exchange matrix extraction in SP. And then there's also ways to automate that, because the more samples you do, we're talking about process that last for 8 hours just to extract, depending on the volume that we're talking about. And then the other issue, it's for PFAS, we're trying to measure at trace concentrations, right? We're talking about parts per trillion. So we really need to concentrate, and that's where SP, and then after that, nitrogen blowdown helps us to concentrate that, both to concentrate and hopefully and get rid of some other matrix interference. And then we resuspend, we send it to the LCMS, and then the LCMS, it's whole mumbo jumbo of complexity, right? That's more of everything. It's analytical chemistry, but from the extraction portion of it, it's already, I think, if you get the extraction correct, I think you're into something. And then the LCMS, it's obviously its own beast of it's a beast of its own. But again, you have to play and optimize with this background EPA 1633 method, that it gives you a great starting point, but then you have to optimize based on what you actually have. In our lab, for example, we've, especially for this wastewater type of matrices that are very high solids, it's, should we treat this as a wastewater? And I think 1633 tells you, oh, if you're up to a higher certain threshold of total suspended solids, you treat it as wastewater. Otherwise, you treat it as solids. So it's okay. So how do you dewater wastewater? So we use a lyophilizer, for example, to help us dewater that and make sure that we account for those losses. And the more complex your extraction, the more opportunities for inefficiency. It's handling, balancing out how accurate can we be, and then also accounting for extraction inefficiencies that are unavoidable. It's going to happen. But we need this very sturdy, intricate methods to be able to answer these very complex questions, right? Like, where are PFAS going? How are PFAS being distributed in our water reclamation facilities? Are we actually seeing some transformation happening in biological activated process? Also, it's complex because certain PFAS might be volatile. If you're thinking about your extraction process, it's, am I vaporizing certain compounds? And therefore, may I be losing some of the type of compounds that I might be looking for, or might be potential biotransformation products? So it's very complex. Spent at least 2.5 years just doing a lot of method development for some of the projects that I worked on, and I think it's still a work in progress. I come from an engineering perspective. I collaborate a lot with environmental chemists, so I'm at the interface of being an environmental chemist and an engineer and thinking, what is the solutions, and making sure that our methods can answer those questions.
And I think that's super common in academic labs, where you're starting with a standardized method and then you're working with that, but you're also customizing that for your own needs. And some of that customization is where some of the headaches can arise because you're going off the beaten path, if that's a good analogy to use.
Yeah, and these are methods or this extraction or testing your hypotheses takes hours. And it's a lot sometimes. Other methods, I deal with biological reactors. Those takes months to answer your question, right? But for whenever you're thinking about, it's not for PFAS in particular, it's not like other environmental techniques rely on other ways like spectrophotometry or other, for example, one of the most basic quotation mark environmental engineering markers are COD or chemical oxygen demand. And that's a chemical reaction that it's then based on color change. You measure spectrophotometrically and then you say, okay, this is the concentration. But with this particular compounds, they're so trace level in the environment and then it's not like they're everywhere, they're ubiquitous. So we have to develop novel techniques and novel extractions for whatever matrix we encounter in the environment, which is almost everywhere, which is scary. We're working around it. And then because it's okay, we're talking about methanolic extraction, sonication, centrifugation. It's a lot of manual labor and a lot of manual components until you figure it out and then you can have more replicable or automation steps, right? And then have the SPE, which also takes, depending if, for example, for drinking water, we have to filter through SPE up to 250 milliliters of water. And in other matrices, I've heard people that have been doing up to 7 liters of water, which is crazy. That sounds crazy to me, but you got to do what you got to do sometimes. And then nitrogen blowdown, which it has its intricacies. I've had to train a lot of, thankfully, we have this. If there's a way that I can automate any process and if there's funding, which is a huge thing also, like I will always bow for the automation in that process because especially when you're dealing with like large number of samples, which was the case, for example, for a research project, that it's more of a community engagement project. We need replicability and we're dealing with like large number of samples, which sometimes is not that common in a research lab, right? Most research labs, it's, oh, okay, I have this experiment and then I'm dealing with, I don't know, maybe my time points are 50 or if you're being very nitty gritty. Now I'm talking about sampling a community and then I have to account for field lengths and I have to account for the actual samples and then we have different type of samples. So now we're talking about hundreds, which is not, and then compound that by the time of hours. So we have undergrads that help us for sure. And that's why I take all the help that we can for in terms of automation. So yeah. When I met you, it was through the nitrogen blowdown, which is like a game changer because before I remember we're engineers, we love to DIY. We have our DIY nitrogen blowdown and it blew away my samples once and I panicked and I was like, we need funding for this, guys. We can't continue to do this. And yeah, and little by little, I think we've been able to stay competitive in our grants and whatever, and we've been able to acquire different automation machines that have been amazing for PFAS analysis and also for the scale that we plan ahead to do and things like that. And also it makes it easier for training people because do I really want to be 8 hours in a lab just watching my sample get into the right levels and just making sure that everything is controlled? Probably not. That's the answer, right? I would rather be doing data analysis or reading more about other stuff if I can. But it's like the nature of this method development for PFAS. It's a lot of it's like finding the right customization and a lot of manual labor to make sure that you can optimize as much as you can in these very suboptimal conditions.
If I could ask you an unplanned question, you had mentioned freeze drying earlier. Freeze drying and nitrogen blowdown can be used for sample concentration. I was hoping you could discuss some of the differences where you would use one approach over the other.
Yeah, so freeze drying, it's it's very useful for large volumes. Also, I think it gives you the additional benefit of it's obviously very energy intensive and it's not like everybody has, especially there's different types of freeze drying. Freeze drying at the commercial scale, a lot of people use for freeze drying foods, and those don't withstand such a slower or a lower temperature gradient. But for research purposes, whenever we use a freeze dryer, it allows us to get rid of the water at very low temperatures without, or it supposedly keeps most of the other organic phases that might be there. But it's just removing water. It's not necessarily removing other organic compounds, depending on the programming as well, right? There's modifications you can have. It's based on the freeze point of water. So you can customize that if you have other organic solvents in your matrix. And the good thing is for the most part, freeze dryers allow you to freeze dry a larger volume, right? Which is very important for us, for example, for wastewater. Depending on the freeze dryer size, there's so many customizations to the type of what's it called? The type of containers that you can have. It's also, you can have unlimited volumes, but it's typically used for a large volume concentration. And it also, quotation mark, guarantees you that you're only evaporating or removing the water. So we're just getting rid of the water, not necessarily the other organic phase compounds. On the other hand, nitrogen blowdown is designed for organic solvents. So nitrogen blowdown thrives in using an organic solvent versus freeze drying, it's more for water, right? And then it's also nitrogen blowdowns, it's also for smaller volumes typically. I don't know, I think there could be nitrogen blowdowns with larger volumes, but we typically use maybe, I think the craziest volume that we've ever tried of nitrogen blowdown, it's maybe 50 milliliters, but I don't think it goes beyond that. Versus in the freeze dryer, depending on the volume of your container and the capacity of your pump, because it relies on a pump to hyperfreeze or reduce the temperature very fast. That one, I think you can go up to 4 liters in one container, which is like a weird case. And ideally, you don't want to fill your container more than, it should be half of the container capacity. So if I have a container of 1 liter, I should only be filling it up to half a liter, for example. But it's still half a liter, right? I don't need to, and I also don't want to be using nitrogen for that. Nitrogen could also be expensive depending on how you source your nitrogen. So it's either way, it's going to be expensive, but at least on one end, you can focus on water evaporation or water removal and large volumes. And then for the nitrogen blowdown, you focus on the organic phase removal and typically smaller volumes. And you have a lot to play with, right? Because again, PFAS has taught me a lot about PFAS. I don't know about the other molecules, but at least for PFAS, we assume that most of them are non-polar, or at least the longer chain ones. So we assume that most of them are going to be retained in the freeze dryer process. And then we go through a methanolic extraction procedure because we know that non-polar dissolves non-polar, and then we try to desorb as much as we can with this organic phase solvent. And then that's why nitrogen blow drying works, right?
Yeah, it's definitely two separate use cases, but I think that's a great summary for people out there who are learning about one or possibly both of those methods.
Yeah, I would say probably the nitrogen blowdown is also going to be cheaper than your freeze dryer. I think freeze drying, depending on what your applications are, typically freeze drying it's used for pharmaceutical manufacturing, but it's finding its ways into environmental testing because now we have a lot of complex matrices. And I think there's also an application, even talking from my biology side, helpful for preservation of other biological molecules like live cells, but we freeze dry them and then it's easier for sharing that enzyme or whatever. So it has a lot of applications beyond just concentrating. It also allows you to keep your whatever product more stable. But again, not as cheap. Nothing is cheap, but not as economically as efficient as nitrogen blowdown.
That's a great summary. Thank you. I was hoping
Yeah.
you could discuss your sampling efforts and any of the challenges you faced.
Yeah, again, there's been different type of projects, so I can talk a little bit about the different projects. So I guess the one that it's the most challenging just because of the complexity and the numbers that we've had to optimize for. I think it's the community engagement project that we have. So currently we're working with communities of Southeast Los Angeles to understand what's their exposure level to PFAS through drinking water and other matrices such as dust, which I don't even want to get into dust because dust it's even more complex. But the idea is to these communities are historically more exposed to other environmental factors that might put them at risk, and in particular through drinking water in Southern California, even though I think most of the water that comes to Southern California, I live in Los Angeles at the moment, it's imported from either the Colorado River or from Northern California. And a very few percentage it's pumped from groundwater sources. But this particular community relies, or the water distributors rely on a larger percentage through groundwater. The hypothesis is, oh, maybe because they rely from groundwater sources, they might be at a higher exposure risk of PFAS through tap water. It's very hard to assess where the water came from because I kid you not, Southern California has the most complex water distribution network of the country, and it has a lot of historical roots if you really get into the depths of that. But basically, we had to plan this huge sampling effort, right? So we were collaborating with health faculty and the public health faculty from USC. I'm in the engineering side, but I collaborated with them. And it was great to collaborate with them because I came from the analytical perspective of like, how am I, we're going to do this? Is this achievable? How are we going to be able to replicate these things? Are we complying with EPA 533? Do we want to do 533 versus 537, right? Which is like slight variation of compounds and etc. And then how are we going to sample? What's the best way to sample? Especially for dust was like coming up with a novel sampling, or not necessarily novel, but like something that's not established anywhere. For drinking water, we thankfully had 533. We opted for method 533, which establishes protocols for sampling for drinking water and tap water. And then for the dust, we were like, we're gonna base it on literature and maybe base it a little bit on the soil from method from EPA 1633, but a lot of it's still trial and error of trying to optimize as much as we can our extraction. But we came up with, I think, a good idea of how can we sample dust in a way that it avoids contamination, has replicability, and understands the fact that if you're going to a household, nobody wants you in your house for more than 40 minutes. Thankfully, we have very engaged participants, and they were very happy to collaborate with us. That's like a huge other study that it will be coming in the next couple of months, hopefully. No, I cannot say too much about it, but we'll see when that comes to the public. And for that one was okay. I had to get my brain a little bit out of the researcher world and be more. If I were like a very strict lab, let's say I'm a lab that has to be certified in all. I was a PhD student at a time, and I didn't have all the resources that maybe a commercial lab has, right? So I was like, okay, how can I make my sampling kits as optimal and as consistent as possible? How do I teach the samplers what's the best way? And I write an account for all the issues that might arise in, I don't know, sampling 75 households in different places of LA, and the issues that might arise through the sampling process, and trying to be as consistent and have good training for them as well, right? Yeah. So that was complex because I had to coordinate, when are you going to drop the samples? When am I going to analyze them? I can keep them in the fridge for too long. So getting my brain into that, okay, I'm not only in the analytical side of just me being in the lab and doing extractions, but how do I implement a strategy that it's already outlined, but once you implement it might be different, into something that will be replicated 75 plus times and relies on 5 different samplers and et cetera and et cetera. And that was for sure complex in terms of just the logistics and the management, and then being able to upkeep with a timeline that we have, because to preserve sample stability, you can't keep them for too long, especially for water. So we had a very, I think it's like for 2 months, I was just like, where's the next sample? Where's the next sample? We're just like processing like crazy. Because once you keep them once you do the extractions, they're more stable and you can freeze them. But once you have them in like raw environmental samples, you can't keep them in the fridge for too long. I think EPA 533 establishes that you shouldn't be keeping it for more than 1 month. So we just had to optimize and coordinate based on our schedules and also the participant schedules. That was one complexity, and more on the analytical side. And for that project, it wasn't that much of an analytical challenge for drinking water because the method I would say is as robust as it can be. And I trust the EPA and the people there doing like as extensive a sample as for the methods that they publish. And then the other aspect of it is which is still a challenge, and we're still working with other collaborators and talking to other researchers to see what's the best way to extract from dust and what's the lowest limit in dust. It's wild because anything can be in dust, right? If you use cleaning products in your household, if you have pet hair, and there are other, only God knows what you bring in your house if you're walking with your shoes and things like that. And not to make people feel like you have to be a germophobic or anything, but it just from the analytical standpoint is you're gonna see a lot of things that you never like that are not PFAS. And how do I manage that interference for my goal, which is to measure PFAS? We're starting obviously from 1633 soils and doing methanolic extractions, and it seems from the research community that seems to be the standard approach. But I just want to make it better because I feel like from a researcher, it's like, are we satisfied with this detection limits, right? There's a trade-off of how well can you extract with your detection limit. And if I'm not reaching as optimal of an extraction process, then I might not be able to detect limits that we might need for PFAS, especially if we understand that they might be at parts per trillion. Because if I can measure it in my control, I don't know, parts per billion, maybe that's not good enough for what it's actually in the environment. A lot of work to be done in that space still. We are hoping that we can get started with a finalized method there. And we're basing a lot of the research that has been published in that space, which is not a lot. And yeah, and then from the biological standpoint for this novel or emerging biotechnologies and the wastewater, right? We didn't get a lyophilizer until I think maybe a year and a half ago, so we didn't have the best capacity. And the good thing about biology, or not the good thing, but I planned my experiments so that for a certain type of experiments, my issue was not concentrating, but actually diluting, because I'm really just putting a lot of pressure on the biological systems. And then, but I know we're collaborating with researchers at the University of Buffalo that have more expertise in the non-targeted analysis. It's a challenge whenever you talk about volatile compounds, but we were able to, again, we design our experiments so that we wouldn't be so much focused on reaching low detection limits for the compound of interest, but rather on seeing like more on a qualitative question of what else it's there, what else it's being transformed in these technologies. And that's where the liquid chromatography mass spec or the high resolution LCMS comes into place to see, and a lot of data analysis, extensive data analysis and data curation to assess, oh, are we seeing this other transformation products? Is this a viable solution? Can this explain what we're seeing in upscale systems like water reclamation facilities? And yeah, I would say be careful with that FTOHs, lessons learned. Those are, again, there's every flavor and taste of PFAS. But for the ones that have this functional group that might be conferred more volatile or semi-volatile properties, it's going to be particularly challenging to extract and account for the extraction losses. And we're still working on it, a lot of method development. But for the other compounds that we've been able to test in other emerging biotechnology studies, I think it's exciting to see what it's possible. And we needed this very high resolution technique and this very intricate data curation and data analysis to be able to see something that we don't account in targeted studies. So it's exciting, but it takes a lot of preparation, never in my life I thought as a environmental engineer and coming more from the biology perspective to understand all of these environmental chemistry challenges that will unavoidably come as part of more complex and more interdisciplinary questions. So I think chromatography will be and is a tool for researchers, whether you're in academia or wherever space you are, for you to really understand and ask the right questions and be very nitty-gritty about, is my question really targeting everything that it's possible out there? And that's the goal of chromatography, is to expand, look at the molecular level, what else is possible. Or are we measuring everything that we need to measure? And then from the engineering side, in my case, building systems that might be able to answer those like molecular chemistry questions. In my case, PFAS.
Would you be able to build on that and talk about why LCMS was the right type of analysis to use for your work?
Yeah, so whenever we think about PFAS, again, sticky molecules, they're everywhere, and they are particularly challenged to measure with any other technique or analytical chemistry technique besides liquid chromatography mass spectrometry or GCMS for the more volatile or semi-volatile compounds. Um, Whenever we think about environmental chemistry, we have, I think chromatography is the gold standard for most environmental sample analysis whenever you think about environmental chemistry. You have ion chromatography, but in our case, none of these compounds are ionizing. You have high resolution LC or liquid chromatography, but those are typically attached to UVVs. So you require your molecule to absorb at a certain wavelength. PFAS do not do that. You also have a spectrophotometrical method. So we're talking about like multiple compounds or targeting multiple compounds, all with different structures. So it will be very hard to come up with a spectrophotometric method that emits a certain wavelength of light and whatever that makes it easier to mix in and look. Maybe a chemist it's out there trying to do that, right? I bet that there's somebody there trying to do that. Then you have, I don't know, ICP-MS. So that's like to measure metals and things like that. And then you have LCMS, which is if all of the above fail or we cannot measure, we go there, which is not the most widely available type of instrumentation in any lab because it's a very expensive instrument and it's also requires a lot of technical ability to operate and to maintain. I call the LCMS we have in our lab Elsa. And Elsa has a personality, and whatever Elsa wants to do, I just manage Elsa. And then you have the GCMS, which might be earlier, or in GC and FID. So you have this separation technique, which is the chromatography part, right? And then you have this detector technique. And in LCMS, the detector technique, it's the mass spectrometry. And within mass spectrometry, you have thousands of flavors of detectors at the end of your pipeline. So for PFAS, because we know that most of them are very nonpolar, and then we know that some of them are, all of the other methods, spectrophotometry or UV-Vis, are not going to be able to detect them. We go to the last resort of mass spectrometry, which is now we have to look at the molecular level, exactly what's the weight of the expected molecule and how would it ionize in this environment so that we can detect and quantify. And then within mass spectrometry, you can be a triple quad, it can be an ion mobility, it can be, there's like multiple techniques, right? But the idea is you separate first your molecule using a liquid chromatography technique. So you use a carbon 18, in our case, we use a carbon 18 or a non-polar packed material, which is carbon 18, and that achieves the separation. So it's based on the polarity of your molecule. And then in order to detect the molecule, you have to ionize it. So ionization is also important in LCMS. And then ideally you want to have a soft ionization technique to preserve the integrity of your molecule if you're using a high resolution mass spectrometry technique, which is what we use. The most common, there's not a lot of different instrument providers. I think it's Agile and Thermo Fisher. We can talk about that in a different topic. And then you have the detection. So in our lab, for example, we use ion mobility, or we use QTOF, or quadruple time of flight, and it's paired to ion mobility if we ever want to decide to use that technique. But that's typically used for protein discovery and biomolecule development. In our case, we're talking about very small in comparison, right? We're talking about smaller molecule sizes. So we have the chromatography that allows us to separate the PFAS based on polarity because we're anticipating these molecules to be more polar. And then in the mass spectrometry, we're able to optimize the mass spec to ionize and travel the ions through a long path, or as long as a path as we can have, to achieve the highest resolution once it reaches the detector. And then now you have two dimensions of separation. You have first the liquid chromatography or the chromatography dimension of separation, which separates your molecules based on polarity, and then you have the more resolved and more accurate degree of separation, which is what you end up using for quantification, which is the mass spectrometry or the mass to charge ratio. So it's both. It's the retention time and the mass to charge ratio. And the cool thing about high resolution LCMS, it's that you can expect accuracy up to, I believe, 1 ppm. So you have exactly the expected mass to charge ratio of your molecule, and you can guarantee accuracy up to, I think, up to 5 ppm variation. So it's, whoa, we have this super powerful machine that obviously it's the last resort if you're thinking about environmental testing, but for PFAS, that's where we are right now, because it's all of the other techniques that could potentially be used are not there yet, or we haven't PFAS, unless we think about other environmental chemistry techniques such as, I don't know, derivatizing the molecule or something like that so that it would absorb to UV vis, and then maybe we can offset the need for a mass spectrometry instrument. But right now, this is where we are, and there's a lot of people that are working both at the government level and in research to think about how else can we measure this thing and how else can we create methods for measurement that are as to the variety of the molecule, of the molecule variations, because right now the only way that we really are able to quantify it's through LCMS, which is obviously a very expensive type of instrumentation and requires all of this maintenance and training without even thinking about the sample preparation, right? Sample preparation, it's one thing on its own, and then whenever you think about the detection method or LCMS, or the analytical, I don't know how to classify them, extraction versus detection, right? Whenever you optimize your detection method, then there's so many routes you can go and to optimize and further expand what you can see with it. And it will probably need in the future a lot of more interdisciplinary research. I know that there's people that, especially for non-targeted analysis, so things that we might not require. The goal is to not require standards for detection, and this is more on qualitative type of assessment. It's very computationally intensive, and we need help of algorithms to help curate, and you need the chemists or people that are trained with that chemistry background to understand, oh, this is a molecule, this just background, this is trash. But ideally, those algorithms can also help us differentiate and help us guide our curation. And there's people that are working in that space for PFAS. I think Fluoromatch, it's one of the biggest softwares that are there that are not by any brand of instrument or whatever. It's like an open source type of non-targeted analysis platform, but it still requires you to do data curation and have an understanding, right? So these are not, I buy a test, I did my thing, and then I measured how much of it is. It's a very intricate technique that requires a lot of training and dealing with the personality of your instrument sometimes, like Elsa for me, right? So I've gotten so many troubleshooting with the instrument that I prefer to see it from the lighter side and just laugh. But yeah, I feel like anybody working in LCMS or especially high resolution instruments, they understand the struggle. It's like you have a Porsche, you have to give it more maintenance, right? So it's like you have a very complex instrument, so you have to account for the complexity if you want it to help you answer these very complex questions as well.
Could you share some of your thoughts on some of the possibilities for bio-inspired solutions for PFAS?
Yeah, so, these compounds, I mean, if you talk to any other person in the PFAS space, they've been synthesized or introduced to the environment in the 1940s. What it's particular about them is that they are carbon fluoride. They all share the quality or the chemical structure that have this carbon fluoride bond, and they're called pair and polyfluorinated compounds because it's either you have a fully fluorinated tail or you have a pseudo-fluorinated tail or pseudo-fluorinated structure. So whenever I think about biotransformation, I think about, can a microbe eat this? Can a microbe cut through those bonds from the chemical level and use that for energy source or for carbon source? And these molecules, which are 100% human made, nobody can debate you that. In 1940s, right, like whenever they were developed, and it was a huge accomplishment from the chemistry side that we were able to perfluorinate and develop these novel polymers that confer qualities that we were not able to give some industrial materials before, right? So PFAS are surface, they're temperature resilient, and then they're repellent to water and things like that. So they're like almost indestructible. Create something with a purpose to be indestructible, a molecule, 100% synthetic or man-made. And now fast forward almost 80 years, right, from that moment, and we've used that type of molecules for a lot of industrial applications, and a lot of consumer product applications as well. So now they're everywhere in the environment, and we created this molecule that it's supposed to be invincible, and now we're like, can we ask the microbes to defeat them? And it's a hard question to ask because the carbon fluoride bond, it's the strongest in chemistry, hence why the molecules have this like very stable qualities. And then also in nature, there's only 2 or 3 molecules that exist that have a fluorinated carbon, and it's only one fluorinated carbon. So from the biology perspective, we are asking microbes, can you develop or can you evolve in this 80 years to cut through those carbon fluoride bonds, even though you've never been evolved to do that? Can you please do it? Please, we need you, microbe. Please evolve to do that. Like a tricky question, right? Because it's like, why on earth would a microbe try to cut through that carbon fluoride bond? And it's asking a very particular biological machinery to do that because it's the strongest bond in nature, and it's not like necessarily like fluoride is a necessary element for most of its microbes, honestly, for most of biology. However, despite of all of these issues, somebody had to ask the question, can we do this? So there was this long-standing assumption that PFAS were non-biotransformable, hence forever chemicals. However, in 2019, a new study demonstrated through a very specific and niche bacterial strain called Acidomicrobium A6, and I know it by heart, that was able to defluorinate PFOA and PFOS, which are both of the most known legacy compounds or PFAS compounds that exist and are widely distributed in the environment. So when that study came out, a lot of people that I think in the early 2000s were like, let's try it with wastewater, let's try it with aerobic, and they were not able to really see any significant change. They were like, no, these are not biotransformable. Let's not waste more time thinking about this. But in 2019, anaerobic iron-producing ammonia oxidizing microbes, and that could sound like a blurb to a lot of people, but basically bacteria that can breathe iron, able to defluorinate perfluorinated compounds, which was a huge deal, right? We had this long-standing belief that these molecules that were synthetically created, and especially that had that perfluorinated tail, so it's not necessarily available for microbial attack, and plus all of the other constraints that you would require for a microbe to eat it, was able to do it, and it was able to export fluoride. Another thing about microbes with fluoride is fluoride can be toxic to cells. That's why we add fluoride in our toothpaste. Mm-hmm. So it's a particularly challenged biological machinery, but not impossible, based on the studies that are starting to come out. And also, it's asking the question, up to which degree can we expect microbes to defluorinate? The good thing about biological transformation is that it's more energy efficient, lower costs than if you were to do another technique, which at this point, we don't have a lot of options for PFAS. It's either advanced oxidation, advanced reduction, or some concentration techniques and supercritical water oxidation, I think. if we come up with a biological transformation pathway, whether it's found in the environment or found the building blocks in the environment, and then we can come up with a synthetic solution. A synthetic problem might require a synthetic solution. Can we find this in the environment under the we're asking nature to give us a new tool basically that it has never evolved to do that, but there's increasing evidence that there is a toolkit. Now the question is, how good is this toolkit? Can it go all the way to completely mineralizing these compounds, because we are seeing some degree of defluorination or chain shortening, but very small, right? And it requires extreme conditions as well. So it's still at a very early stage and very exploratory stage, but there's light at the end of the tunnel, I like to believe, hopefully after 5 years of research and doing all of this method development. But yeah, it's also changing that understanding that from the biological machinery part, it's something completely strained to nature. Only 2 or 3 molecules have really been able to do that. As we have more tools also from the biology perspective, we can design hopefully other tools that kind of offer us alternatives, because right now whatever we're doing with PFAS, we're basically applying concentration techniques at industrial scales, right, to manage the waste, such as ion exchange or granular activated carbon or other techniques that concentrate the waste and remove it from the water, remove it from whatever we need to clean. But we still have that waste, and we still need to think about strategies to mitigate that waste. Otherwise, it's going to bit us in the tail 20 years from now again, and we're in this endless loop of we synthesize these molecules, and they're so strong that not even us, not even we can destroy them, or not in natural conditions. And we also have to keep exploring from the biotransformation perspective what else is possible. Maybe we needed more time for biology to evolve. It's been 80 years since their synthesis. We've given them enough time, but at the same time, evolution takes time, and then we also have to create these extreme conditions. But there's hope. There's a couple of studies that show the enzymes that are able to defluorinate, at least for compounds that exist in nature, like fluoroacetate, for example. With more advanced technique, we can really unveil this if the answer is there in nature. And if it's not, show must go on and we will have to keep trying. But I think there's opportunity there. And a lot of my research was like, yeah, seeing what it's possible with the microbes, what it's possible at the biology level, which required me to acquire all of these environmental chemistry tools, because it's an interlinked question. You have to understand the chemistry, you have to understand the biology. In the biology, it's giving us some very PPT light, parts per trillion light, but it's giving us something. And I'm excited to see where it will go. We're still at the infancy of understanding what is possible with that. And that's where the chromatography tools will help us, right? Especially the very advanced non-targeted screening or the more optimal extraction technique.
I know that a lot of your work is still in progress, but is there anything you'd like to discuss regarding your upcoming work for 2025, or just anything else you'd like to share with the audience?
Yeah, so yeah, you will see hopefully more papers coming in terms of biotransformation and that community engagement work. We're still finalizing some of that work with the community, so that's why I didn't want to talk too much about the results. And then for the biotransformation, I think there's a community out there that it's still, regardless of all of these challenges that you're like, why on earth would you try to biotransform these things? I think it's exciting, and it offers us to think about solutions that are more interdisciplinary and for sure are going to be more complex, right? More complex than what we have with the status quo. So I think most of the papers that will be coming out this year are going to be talking about that and talking about the microbial world and what were we able to explore, especially about the anaerobic digestion world, which is a lot of what my research lab does. We love aerobic stuff. We breathe oxygen. What about the microbes that don't, right? What can they offer us, and what solutions can they offer us? Can they offer us solutions for PFAS? Can they offer us solutions for mitigating other, contaminants of concern? It's still, I think most of the work is still at the exploratory phase, but it lays out the foundation of what happens at the anaerobic digestion microbial world and with as it interfaces with PFAS. And I'm really excited to have been part of that foundation laying and be like, I sound a little bit crazy for even trying to do this. My advisor and I were like, are we sure we want to do this? It's again more of exploratory, but I think it's yielding us. We can't give up because this is a very wide stream, very mainstream problem that we need to think about all the possible solutions beyond just chemistry and physical-chemical methods, which it probably the solution, it's not necessarily just one or the other, but the best of everything because it's a very complex problem, particularly for PFAS. And I think biological tools offer us that new opportunities. And with a lot of the technology that it's showing up now, sequencing technologies, but that's more on the biological standpoint. And then the chromatography technologies, right? And we need still chemists that develop novel and more efficient methods and better. And now we also have to think about AI, right? Like, whoa, AI even wasn't even part of the conversation before, but maybe AI gives us a tool for molecule discovery, for example, for non-targeted analysis for PFAS, for example, that might be useful. And then we have this huge databases of, or we have to keep mining what it's possible. We can just the status quo sometimes, especially when you have such an urgent and such wide, wide mainstream problem that it's affects everybody at this point in the U.S. Not at the concentrations that are concerning. I think that's why PFAS has been a little bit slow in its regulations. But I think the question it's like, for how long can we sustain this constant exposure? Up to which point it's PFAS gonna be? Can we sustain this like constant exposure of PFAS, and up to which point we're gonna see human health impacts that might be associated with them at the general population level? And this is not to be fear mongering, but I think it highlights the need to evaluate everything that it's possible to solve it, even biological transformation. And that's where my space grows and expands.
Thanks, Bianca. I really appreciate that time today, and thanks for sharing all of your research with our audience.
Yeah, thank you so much. If you're dealing with PFAS or measuring them, which I understand it's a very long and challenging process, don't feel alone. Everybody doing PFAS work will understand you and will understand the pain. And yeah, think we're moving slowly but surely. And hopefully, together as a scientific community or wherever you are in your research project, we can hopefully debunk that idea that they're forever, whatever shape or form that takes.
Awesome. Thank you again. I appreciate it.
Yeah, thank you.